Good evening, everybody. Can you hear me okay? Yes, great. So it's my great pleasure to be your entertainment tonight. I'm going to tell you about how I think advances in miniaturization might impact human health. I'll give you a couple of examples from our work in cancer research and a couple of examples from our work in liver disease. And please feel free to interrupt me as we go with questions. So in the space that used to house a single transistor, we can now fit 1 billion. That made it so that a computer the size of an entire room now fits in your pocket. And that portability enabled the inventions of things like smartphones and Fitbits and GPS tracking devices. So you might say the future is small. As an engineer, I find this miniaturization revolution inspiring. And as a physician, I'm incredibly optimistic about what miniaturization might offer human health, in particular in the field of cancer. So it turns out that there's an incredible opportunity to save lives lost from cancer due to the early detection and prevention of cancer. Worldwide, over 2/3 of cancer deaths are actually fully preventable today using techniques that we know about, things like vaccination, timely screening, and of course, stopping smoking. But the early detection of cancer is problematic. Even some of the best tools cannot detect cancer for up to 10 years after it started growing, when the cancer is more than 50 million cells strong. So what we've been excited about is what are the opportunities for miniaturization to help us possibly detect cancer earlier and save even more lives than we can today. Here's an example. On the left, what you see is a standard microscope that you'd find in a pathology lab in a hospital. And this would be used for looking at a cancer sample or, for example, a biopsy. So this microscope costs $7,000. This is state of the art. And on the right, what you see is that this whole part has been miniaturized into this $10 part by one of our alumni, Rebecca Richards-Cordham. She's now at Rice University. This $10 part fits on the end of an optical fiber, and it can be brought to the patient instead of taking a sample from the patient and sending it to the microscope. So this allows a decision to treat to be made in the moment. Okay? So this is one thing that miniaturization has to offer us. But if we miniaturize this part even further, we can start to think about a cancer detector that could circulate in your body, find cancer all on its own, and send a signal to the outside world. Now that sounds a little like science fiction, but actually I'm going to show you that we think it's possible. So just to give you a sense of scale, what I'm going to be talking about are nanoparticles that are 100 nanometers in diameter. These particles are 1,000 times smaller than the width of a human hair. And at that length scale, what we call the nanoscale, these materials change how they travel in your body. Okay, so here's a little movie to show you what I mean. This is a blood vessel in the body, and around the blood vessel is a tumor. And we're going to be injecting the nanoparticles into the vessel. What happens is that the blood vessels of many tumors are leaky. And so nanoparticles can leak out depending on how small or big they are. So here you can see the 100 nanometer particles are leaking out, and the larger 500 nanometer particles are staying behind. We decide some of these 100 nanometer nanoparticles to be sensitive to chemicals that tumors make when they're invading, called enzymes. Each one of these enzymes can activate a nanoparticle and create 1,000 fragments in an hour. In engineering, we call this an amplification reaction. So we've made an ultra-sensitive cancer detector that's 100 nanometers in size. Okay, now we'd like to get that signal outside of the body. It turns out that the kidney is itself a nanoscale filter. So here what you're seeing is that things are coming through the bloodstream and coming out of the kidney. The 100 nanometer particles stay behind. But these smaller 5 nanometer fragments come out in the urine. So the kidney is a 5 nanometer filter. So if we put this all together, I can make 100 nanometer particles. They can leak into the tumor. They can be activated by the enzymes. These fragments can be released that are small enough for the kidney to filter out. And now they end up in the urine. Okay? So I've injected a cancer detector, and now I have something in the urine. The urine is the outside world. That is some place that we can detect it. So how do we detect something in the urine if it's so tiny? Well, it turns out that these are just molecules that we've engineered. And because they're completely synthetic, they don't look like anything else in your body, we can design a test to detect them in the urine. And we've been very interested in this kind of test. So this is a class of tests that's being developed now in the field called a paper diagnostic. And you'll know it because it looks a lot like a pregnancy test. It's a paper test. You pee on a stick, and the test lights up. So there are more and more complicated paper diagnostics coming, and we've designed one to detect those molecules that came off the cancer detector in the urine. So the vision for this technology is that you would give a shot, you would wait an hour, and that you would do a urine test on a paper strip. And this could potentially allow us to detect cancer in patients earlier than 10 years after it started growing, and save even more lives than we can today from screening. This technology is the basis of a new startup that we've just spun out of MIT. It's called Glimpse Bio. It's just 3 months old, and it's just getting started, but this is where I hope it will go someday. So this is what we're hoping we can do for early cancer detection. But what about treating cancer? There's another idea that we and others have been working on where you could take these same nanoparticles and you administer them to a patient. This patient has a brain cancer. And the particles would be packed with chemotherapy drugs. Okay? And the idea is that the particles would avoid the filtration organs of the body's liver and spleen, which are the natural filters, and be able to accumulate the drug in the tumor and deliver more chemotherapy to the tumor and spare the normal tissues. Okay? And we call this targeting. Now, that's actually a lot to ask of a particle. That particle has to find the tumor and deliver the payload. But this is something that the field is working on. In fact, there are MIT startups in this area. You may know of Bob Langer's recent startup, which has since gone public, called Bind Therapeutics, along these lines. So what we've been very interested in is pursuing the same concept with materials that have their own properties at the nanoscale. So I told you before, if I shrink a material down and I put it in your body, depending on how big it is, you can control where it goes. We call that nanoscale biology. But what else happens when you shrink materials down is their physics changes. Okay, let me show you what I mean. This is an example. Let's see if this movie will play. Touching this little thing. Well, I suppose I could describe it. Why is this so hard? All right, I'll describe it. Except for it feels actually like I'm so close. Right there. Oh my God. All right, well, if anyone's a computer whiz, they can come up here later and we can play the movie. Well, I'll describe it for now. What happens at the nanoscale is you take a material like gold. Gold is optically gold. If you shrink it down to the nanoscale, it turns red. And if you change the shape of the material, are you going to try? Yeah. Change the shape of the material. It will change the way it interacts with light. So what's interesting about that is if I take a Oh, thank you. There's the gold shrinking.
Yeah, we're not going to go full view. But
Starting out red, 100 nanometer gold particle. Now we're going to, thank you, elongate it. We're going to elongate it into a rod shape and shine light on it. This particle is going to heat up because the electrons on its surface are so mobile, and it's going to heat up depending on its shape and the light that you shine on it. So the more elongated particle was sensitive to red light, and this shorter, fatter particle is sensitive to green light. We call these materials plasmonics. Now, if you shine these wavelengths of light on a brick of gold, it won't happen. This is a nanoscale property. So they become antenna for light at the nanoscale. And the red sensitive particle is about 50 nanometers long and about 10 nanometers wide. So I have a gold particle that is an antenna for near infrared light. What do you know about near infrared light? It can penetrate through tissues. It's what we call the optical window. It's about 800 nanometers. It can't penetrate all the way through tissues, but it can penetrate further than most other wavelengths. So as engineers, we really like this wavelength of light for exciting materials. So we wanted to use this idea. Let's go back to my, let's see here. Yay. We wanted to use this idea to create a targeted chemotherapy system that could deliver more chemotherapy than just a nanoparticle on its own. And so what we did was we made a two-particle system. In this system, the gold nanoparticles are the blue guys, and all they have to do is find the tumor. They don't have to accumulate very much. They just have to find the tumor. They don't have to carry any payload. But when they get there, we shine light on them, and they get excited, and they send a signal. And then we send a second nanoparticle, a red nanoparticle that's carrying a lot of the payload. And it's carrying the payload, and it's listening for the signal. And it's a molecular signal, for those of you who are interested. So it's a two-particle system. The gold nanorods send a signal. The red nanoparticles carry the payload. And together, what we find in models of breast cancer is that we can get a 30,000-fold amplification in the amount of drug that we get deposited using this strategy. So instead of just using one particle that has to find the tumor and carry the payload, when you divide up the job between find the tumor, send a signal, and carry the payload, you can get a huge amount of amplification. So this is one way we think. This is a model of breast cancer in mice, and you can see that these tumors are shrinking, whereas even the same two particles delivered in that same animal model of cancer continue to grow. So it's only when they can communicate that they shrink. We're pretty excited about the possibilities for this. Okay, so here I want to take you on a little tiny side diversion, which is a sort of quintessential MIT story about 3 students in my lab who saw this project and had an idea for a startup, which has started in 2011 and now doing extremely well, called Sienna Labs. And it's a little bit unexpected, which I think is the best kind of startup story. So this student, Jeff von Moltzon, a student in the lab, he was doing the work that I described to you. And so when he first started doing this work, he was taking these gold nanorods, these antennae, and administering to the animals that had tumors in them and shining light on them. And here he's taking a thermal image, and you can see that the animals that had the nanorods, the nanoantennae, were heating up, and the control animals were not. So we published this paper in 2009, and Jeff went on to win the Student Lemelson MIT Prize and become a venture capitalist at Flagship Ventures. And he's since started a bunch of companies, and he never touched nanorods again. But 2 of his lab mates, pictured here, Todd Harris and Alice Chen, had been in the lab with him at the same time. And Todd went off to McKinsey, and Alice went off to a startup in the Bay Area. And the 4th person in this story is not pictured here, is Todd's wife, Ashley. Ashley is blonde, okay? And this will become relevant in a second, okay? Which is, Ashley came home one day and said to her husband, Todd, my former student, I cannot get laser hair removal because I'm blonde. Okay, so let's just back up for a second. What is laser hair removal? Laser hair removal, you're looking at a section of the skin. Okay, and the way laser hair removal works, for those of you who don't know, is if you have a dark hair, it's pigmented here, it's in the skin, and you shine near infrared light on that dark hair, it will heat up just like a dark car does on a summer day, more than a white car. Okay, and it will heat up and it will damage the hair follicle, and that hair will no longer grow. Okay, and this is the basis of a very large dermatology industry. So Ashley came home complaining that she could not get laser hair removal. Why not? Because she's blonde, right? It doesn't work with blonde hair or gray hair because you have no contrast. So Todd remembered that he had been in the lab with Jeff, and Alice was also there, and he called them both, and he said, I think if we make a cream of plasmonic nanoparticles and put them on the surface, they are small enough that they will fall down this hair follicle, and we can create the contrast that the hair doesn't have. So that's what they did. So that became the basis of Sienna Labs. This is about a $40 million company right now. They're in clinical trials. And they have a cream. I'll show you how it works on the next slide, which is plasmonic nanoparticles, and they shine. You put them in, and you can shine light on them, and they damage the hair follicle. Okay, so that's dermatology application. I'm a medical doctor, so I was interested in what else could they do with that. So what else could you possibly do with that? What else do you see here? What's this? This is the sebaceous gland. This is the source of cystic acne, right? Acne is something that you know it can be damaging to the skin. There's one treatment, systemic treatment for acne called Accutane, which has a black box label from the FDA. Okay, and so what Todd and Alice and Jeff realized after starting Sienna was that you could use this not just for hair removal, but also for cystic acne applications, because you could get the plasmonic nanoparticles into the sebaceous gland and you could damage it too. Now, what about the fact that we use gold? Gold is not a material that we think of as particularly cheap or degradable. But what Todd and Jeff and Alice realized was that these plasmonic properties of these metals are shared amongst other metals. And in fact, silver has a very similar property, and silver has been in people for a long time in bandages and all kinds of other applications. So they started Sienna Labs. These are the particles they make. They're silver particles. They're coated with a layer of silica, so they're biocompatible. This was their first clinical trial. This is a cream and an applicator and a laser in the dermatology office. And they've now done 20 patients for hair removal, and they're about to enroll about 90 patients for cystic acne. So it has nothing to do with cancer, but it just goes to show you that you never know what happens from your friends in graduate school. Okay, so I've told you about cancer, I've told you about a side diversion, and now I want to turn to a little bit about talking about growing regeneration of human organs. And by way of introduction, I want to say that, I started my talk saying that miniaturization is very powerful. Then all the examples I gave you were what we call the nanoscale, that kind of 100 nanometer and smaller. And if you look at this kind of ruler up here, 100 nanometers is way down here. But actually, it turns out that the miniaturization of electronics, semiconductor manufacturing, really has given us a whole world of tools, even beyond these nanoscale tools. So there are all kinds of things that we learned how to do to print circuits on silicon to make our circuits faster and more integrated that can be incredibly useful for medicine, even larger structures in the nanoscale. And what's really exciting to me, trained as a doctor, is that if you look at a tissue, if you look at a piece of tissue, so this is a piece of the liver, each one of these is an individual liver cell. Actually, it turns out that your body is organized at about this same length scale. So each cell is about 10 to 20 microns. Clusters of cells are 100 microns. These red blood cells are 7 microns. The functional unit in the liver is about 500 microns. So if this is your kind of toolbox, and this is the thing you're interested in, it turns out to be completely compatible. There's all kinds of things that you can do to study these tissues and try and heal these tissues using this world of tools that have become available. Okay, so let me give you some examples. So we're very interested in the liver. The liver is my favorite organ, my first love. And this is what it looks like in your body. Who knows what the liver does? Filters out toxins, it detoxifies. It makes bile for digestion. It does all kinds of metabolism, sugar, fat, and protein metabolism. And it makes proteins. So it makes like your blood proteins, clotting proteins, albumin. So it actually has 500 different functions. It's essential for life. There is no treatment for liver failure short of whole organ transplantation currently. How does your liver fail? Cancer. Injury from alcohol or from drug injury. One more way. Two more ways. Hepatitis. So hepatitis C. Hepatitis C is amazing. It's been an amazing time for us liver lovers because this disease got cured. And it's very rare in your career that you see over a couple of years that this disease is now cured. It's expensive to cure, but it's cured. Hepatitis C, hepatitis B. And then one more, we call it the stealth epidemic. Autoimmune disease wasn't on my list, but that is a problem. It's a smaller set of patients, but it is a problem. The big one, 40 million Americans have fatty liver disease. It's connected to diabetes. So it's driven by type 2 diabetes and obesity and metabolic syndrome. So first your liver gets fatty, and then it gets inflamed, then it gets scarred, and then you're at risk for cancer. So 500 million people have liver disease worldwide. It's a huge problem. And like I said, the only thing to do is to do a transplant. So we've been very interested in this for a long time. And the first thing that we were interested in, just as scientists, was why is the liver organized in this way? And I won't describe all the ways that it's beautifully organized, but you just observe it's beautifully organized. We were interested in studying some of these human diseases that really there were no good animal models for in a petri dish. By using human liver cells, if we could manipulate them using some of these fabrication methods that I mentioned. And I'll show you an example. And then we were interested in coming up with an alternative one day to transplantation. Wouldn't it be great if you didn't have to wait for someone to donate a liver to get a liver? And we're getting there, but we have a long way to go. Okay, so let me show you what I mean. So the sort of central player in all those 500 liver functions is this guy, what I called the liver cell before. The fancy name for it is the hepatocyte. And the problem with this cell is that when you take it out of that gorgeous architecture and you put it in a petri dish and you look at it and you say, okay, how are the 500 functions doing? They all do this. They all crash. Because this cell is very sensitive to what we call its microenvironment. It's a neighborhood. What other cells are around it? What other scaffolding is around it? What other proteins is it seeing? And so when I was a graduate student here in the 1990s, Marty Schmidt, who's now the provost, was on my thesis committee. I worked for Mehmet Toner at Mass General Hospital. And I went over to the microfabrication facility in the EECS department. And we decided that we could recreate a friendly microenvironment for these liver cells using some of these semiconductor fabrication methods. Okay, that was the basic idea. And the method that I came up with is shown here, where what you do is you make a little stencil just like you use in grade school that you draw your alphabet through. But this is a stencil, it's a plastic stencil, and through it we can pour proteins that the cells are going to stick to. And in the end, we could get these little patterns of liver cells. So we had created 250 cells packed on this spot, and we could surround them with neighbors that they saw in the liver. And these two manipulations of patterning these cells and giving them friendly neighbors allowed them to stabilize, whereas before they would die. Okay, so now we had a little human liver, we call them microlivers, in a dish. And we got really excited about that and thought about what we could do with it. So we started a company in 2008 called Heprogen. And this company manufactured these They just turned into Ascendance Biotech this year and sells them to pharmaceutical companies all around the world. Why would you use a little microliver if you were a pharma company? To predict the safety of your drug, because it might be toxic in the liver, and if it's a liver drug, to see if it works, right? Safety and efficacy. And then finally, drug metabolism. We talked about detoxification. Your liver is actually the organ that gets rid of all these drugs. Okay, so this was very exciting, and we've gone on to do some interesting things in the lab since. So this is another sort of frivolous but interesting project. This is hepatocyte wallpaper. It's part of a traveling art exhibition called Colonies. It was done by this postdoc of mine, Tal Zanino. He just joined the faculty of Columbia. And an artist in residence at the Media Lab last year named Vic Muniz. He's an amazing Brazilian artist. And he likes to make art that has kind of ensemble properties. So you look at it and it's a beautiful image, and then you zoom in on it and it turns out that it's made actually out of interesting component parts. And the component parts for him that were interesting were our liver cells. And this was very easy for us to do because we had already figured out how to use microfabrication to pattern the cells. So we could do anything arbitrarily that he could give us. We could make these beautiful patterns. This is a project that we did together with the Gates Foundation, where we made this beautiful image which looks a little bit like a tapestry. And actually what it is liver cells that have been infected with a virus, which is the basis for a common vaccine. And this project, which the Gates Foundation started, was to try and use art to shift the dialogue about vaccination, to have somebody approach this image just as a beautiful image, and then to teach them what it is and what the value of the underlying science was, to sort of engage them in a different way. So all kinds of sort of pretty things that we can make with this microfabrication. Okay, but what can we do that's useful? So the sort of poster child for hard to predict drug toxicity had been this drug. This drug was a hepatitis B drug in the 1990s. In the safety trial, in the safety phase of testing, when you first administer the drug to patients and you give, it's called dose escalation, so a little bit more, just make sure it's safe. 5 out of 15 subjects had liver failure and died. This was a horrible result. And looking back on it as a community, we could not have predicted it. It didn't show up in culture, it didn't show up in animals. The FDA agreed it couldn't have been predicted. And the reason is that animals are not people, and their livers metabolize the drug differently. So what we showed, we were very excited to see, was that in these human microlivers that I showed you, that in fact, today, you could call that toxic. And so this is why groups are now using this for their new drugs to make them safer. So this was very exciting, and as I said, the company has gone on to do this. But then we got excited about other diseases of the liver. So let's just see if I do your trick again. I think I might have to. So it turns out that liver infections are, where's my friend from the audience over there? There we go. Turns out that there's a lot of liver infections that are difficult to study. And this one is one that we don't think of as a liver infection. This is malaria. So an infected mosquito bites you. The parasite goes to your liver. It sets up shop in the hepatocyte. It grows there silently for a week. It amplifies 10,000 times, and then it bursts out into the blood to give you all of the symptoms of fever and cyclic fever of malaria. Then what you're seeing here is a naive mosquito will bite that person and then transmit the parasite further. So malaria, as you probably know, kills 700,000 people a year. Most of them are children under the age of 5. And there has been a great interest in finding drugs that could kill the parasite at that early liver stage, before that amplification happens, before the patients have gotten sick, before the next naive mosquito could catch malaria. Okay? But this has been challenging. There's only 2 drugs that kill the parasite in the liver at that stage. And one of the main reasons is that we can't grow it. You can't grow the parasite in the liver stage to kill it. So about 5 years ago, we started working with the Gates Foundation to use our human microlivers to infect them with malaria. And our idea was this: if this is the normal life cycle, the mosquito, the liver, it's growing, it's amplifying, and it bursts into the blood, we should be able to do all of this part outside of the mosquito in the lab. And if you could do that, then you could have a petri dish, you could dose it with drugs, and you could see if your antimalarials would work or not. So we've done that now. We've taken these little microlivers, we take the parasite. Where do you think I get this parasite? Mosquitoes, but there's no malaria in the United States. So my team has to fly all over the world. The latest, sort of most robust source of Vivax malaria, which is a chronic form of malaria, is Thailand. And so this is what we do. We wait for the rainy season. We get a call from our Thai collaborators. We fly all of the pieces of our liver parts to the Bangkok airport. We've learned what not to call our supplies. Like we don't call them drugs because they don't clear customs. We call them compounds. And we assemble the human microlivers there. They wait for patients to come into the clinic. They draw blood from an infected patient. They have an insectarium where they feed the mosquitoes the blood. We wait for a week. The mosquitoes grow the parasite in their salivary glands. We collect the mosquitoes. We cut off their heads. Just a mosquito. We collect the parasite, and we dose our cultures, which we've now assembled on site in Bangkok. Okay, and we wait a week, and they grow 10,000 times. And as they're growing, we can test drugs against them. So this is very hard work, but it works. These are the two most prevalent forms of malaria. Took us 5 years to get this working, and these are the two drugs that I told you about that exist on the market in patients today that have liver stage activity. And now we're working with the Gates Foundation to test novel drugs to help them prioritize them for clinical trials. So we're really excited about that, even though it's such hard work. In the last few minutes, I want to just tell you about how we're thinking about going from these little tiny livers to the kind of grand challenge of building a big liver for a patient. So the first question you'd ask if you're quantitatively minded, which I know all of you are, is how big does it have to be? So how many liver cells do you think we need to make a liver, to support a patient? 100 liver cells? More. More. Yeah. So the liver has 100 billion cells. And we know from transplant data, from some trauma data, from some pediatric data, that you need probably 10 to 30% of the liver to support a patient. Okay, so 10 to 30 billion cells that I have to build if I want to replace a transplant. That's a tall order. And I'll tell you in a second that instead of just building it, we're trying to harness biology to help us grow it as well. But when we got started in this business, this was pre-3D printing, we were inspired by an old manufacturing technique called stereolithography. This is the precursor to all of the 3D printing that you've been hearing about in the lay press. And the way it works is you have a vat of a plastic that's going to harden when you shine light on it. And if you shine light on this layer by layer by layer, you can build a complicated part. And we got started in this in about 2002. We thought, wouldn't it be cool if we could build livers this way? Because I told you we had been building livers on surfaces with light using microfabrication, and we had made the microlivers. So we thought, okay, we could use the same kind of approach. We could build a three-dimensional liver with all that gorgeous architecture. So that was a great idea. The professor always has like that bright idea. It took a whole PhD thesis to get working, 5 years. But my first graduate student that got this working, Valerie Lu Sang, got these liver cells working in this particular plastic, and she could make all kinds of interesting liver parts with light. This is one of my favorites. These we call liver beads. This is a little microfluidic device, and the plastic is coming in here, the polymer solution, and you can see these individual liver cells. And they're budding off into these droplets because of the way the device is designed that are all exactly the same size. And then we shine light on these droplets, and we can literally turn this thing on and make buckets of these beads. Now you have little parts of liver that you can assemble into something bigger. This was the simplest thing we assembled in the beginning. This is what we call our contact lens liver. You can see why. It has about 1 million cells in it, okay? So it's 3 or 4 orders of magnitude too small. But we were starting small. And so we figured out now that we could get the liver cells to live in this part. But what else do you think you need to build a big organ? You need blood vessels, absolutely. You need nutrients, right? So if you get further than 40 microns, 100 microns away from a blood vessel, that's too far for the oxygen to get there, right? So you could build a huge organ, but unless you build a blood vessel architecture with it, none of them are going to live. So we needed to build blood vessels. So in order to do that, we collaborated with another MIT alumnus named Chris Chen. He's now at Boston University, and he's an expert in building blood vessels. And he and I had been friends since HST days. We started our labs at the exact same moment. We used to run a joint lab retreat every year. We used to read each other's grants. And I would say about 7 years ago, we said, well, let's do something together. Let's collaborate. And he said, well, I build blood vessels. And I said, well, I build livers. I said, okay. I was thrilled to vascularize liver. So that's what we did. So what he's been doing is using 3D printing in a different way. He's been using 3D printing to build blood vessels. Okay, that's a little counterintuitive, right? Because a blood vessel is a space, right? It's not a structure, it's a void. And so what he did was actually to print a network of what we call something sacrificial, something that would dissolve away. So the thing he prints is where he wants the blood vessel to be. And then he surrounds the blood vessels to be with the polymer, and then he dissolves away the network. So let me show you what I mean. Oh boy, again with the movies. I think this one might work without me. Oh, there's sound.
This is a RepRap, a popular open source 3D printer in the lab at the University of Pennsylvania. It's being used to print blood vessel networks out of sugar for research.
Okay, I can't stand that guy's voice, I'm sorry. But this is really all you need to see. So we're printing this sugar network with a 3D printer, and then we're surrounding with the polymer, and then the sugar dissolves away, and it makes blood vessels inside the body. Okay, so he makes the blood vessels, we surround this with the liver cells, and then we have vascularized liver. Okay, so the last piece of this puzzle is I told you we started with 1 million cells, and we need 10 billion cells at least. So if we're going to print this, it's going to take a long time. Right, so how might we scale this up? It's an engineering problem. Yeah, so one way is to take those beads that I talked about, right, and to assemble them, to sort of print them all offline and then assemble them in some larger structure. That's the engineering scale-up way. But these are living structures, and they can grow. And in fact, the liver is a unique organ. It can regenerate. And this is sort of, there's an old myth about Prometheus stealing the fire from the gods. Does anyone know this story? He gets strapped on a rock as a punishment, and every day an eagle comes and eats his liver, and every day the liver grows back. And in the liver community, we say, oh, people have known since ancient times that the liver regenerated. And in fact, the liver can regenerate. So you can regrow 70% of your liver mass in a few weeks. And it's the basis for a very interesting procedure, which you may have heard about, called split liver transplantation, where you can donate a part of your liver to a child. Your liver will grow back. The child's liver will grow over the course of their life. So it's a very unique organ, and it grows this way without a stem cell. It's the mature hepatocyte that does all those 500 functions that makes it grow. So we wanted to see if we made one of these livers with Chris, and then we gave it a regenerative stimulus, whether it could grow in the body, right? And that way we wouldn't have to build all 10 billion through scale-up manufacturing. We could build part of it through scale-up manufacturing, and part of it could grow. And so this last summer, we got our first results, which I hope you can see here, where when we put this little liver in an animal and gave it a regenerative stimulus, this liver grew about, so far, 50 times. But it grew in the body with no engineering at all. So we're pretty excited about this because now we have some scale-up manufacturing tools and we have some natural regeneration tools. And together, we think, eventually, we'll be able to build this liver for patients. All right, that's all I had to tell you today. I've sort of touched on a lot of these concepts, but really there are more and more tools just pouring out of the community, out of the engineering community, that are really exciting and available to borrow for medical applications. And I sort of think this approach that I've talked about can really be mapped onto many other diseases and organ systems of interest. We ourselves are working on other diseases as well, but mostly we work on liver disease and cancer. And the idea is that we can model human tissues, we can monitor disease better, and we hope that we can treat it more effectively. The last thing I want to say is that we need talent in this field, and there's not enough women engineers still. You probably know that 12% of the American workforce is women. 3% of high-tech CEOs are women. 20% of undergraduates are women. So the numbers are low, and they've stayed low. They sort of stopped climbing in the mid-1980s. So we've been working on trying to really increase the pipeline. This is an outreach program I started when I was a graduate student at MIT in the 90s, and it's still running. And my older daughter is now old enough to be a student in the program. It's called Keys to Empowering Youth. She's actually coming to the lab again on Saturday. I'm also the advisor to the Society of Women Engineers, which is an MIT undergraduate club that now run this program. And then I try to do a lot of outreach work. This is the most terrifying talk of my life, which was live on Broadway back in October, and will come out later this spring if you guys want to see it. But I think these are all ways really to show young girls that this is a rewarding profession and that they should choose it and come to MIT. So with that, I'll just close and thank you for your attention. Thank my group who really does all the hard work, and I'm happy to take questions. We do have two microphones. If people have questions, if you want to come up to one of the microphones, we appreciate that.
I have a question. I was hopeful too. I'm wondering, with the tray of microlivers that you had, it was like 24 kids and 50 microlivers each. How much would one of those trays cost at scale, the production scale? And my second question is, does the manufacturer's liver that you guys create, does it know when to stop growing, when it's implanted?
Those are both great questions. So I don't want to speak too much of the cost of the product per se, but it's several $1,000s. But what limits it actually is the cost of the liver cells, because the way those are acquired are from organs that are already few and far between. And then the organs that are not transplanted, we use those for harvesting. So that's a very expensive process. So we're actually trying to grow those liver cells also, because they should be able to grow, as well as use stem cells. So we use iPS cells to create those cells. So the cost of that product should come down. The question about stop signals in liver regeneration is a very hot topic in the field, and there have been stop signals identified. Already we know, though, that there's basically a feedback loop, and so when the liver grows big enough and the proteins in the blood are high enough concentrations that the growing stops.
Could you expand the molecular signaling? I think it was red and blue, if memory serves, back about 12 or 14 slides. So is this some sort of bonding that's occurring? Is something being emitted? What is happening?
Yeah, that's a great question. So I think he's asking about the nanoparticle system.
Correct.
Where the gold nanorods, they actually heat the tissue locally, and they start the coagulation cascade, which is the blood clotting cascade. And then at the end of that cascade, there's a transglutaminase, which is an enzyme in factor 13 that we use to target with a peptide.
Okay. And the other thing, oh man, I glitched. You were talking, yeah, the polymer that
Yeah.
that was forming the, I'm sorry, did you call them cells? Or
Beads, they call them
beads,
beads, but yeah.
The beads are living, yes?
Yes.
Okay. So the polymer is, I mean, is it some aggregated monomer plus some tissuing? What is it that makes that polymer?
Yeah, it's also a great question. So for those of you who aren't experts, a polymer is a chain of repeating units, and these are macromers. So they're polyethylene glycol. It's a very inert polymer, and they have reactive end groups. And so what happens is that we put a photoinitiator, a chemical, in with the cells and the macromer, and then when we shine the light, they just cross-link right at the ends.
But the living part, I mean, is this genetic material or not?
The cells are alive.
Yeah,
So
they're bonded to the
they're entrapped. Yeah, they're molecularly entrapped. So it's a hydrogel. It's 90% water. And then the living cells are right in the mix. It's kind of the consistency of Jell-O.
So it's some salad dressing and a little bit of meat.
Yeah, sure.
Well, polyethylene glycol.
Exactly.
Salad dressing.
Indeed.
Thank you.
I just wanted to ask you about the nanoparticles that you were talking about for diagnosing tumors.
Mm-hmm.
Now, don't they have to be specific for the type of tumor? And how do you know if you haven't diagnosed the tumor already?
I know, it's tricky. That's a great question. So that little yellow chemical enzyme that came out, I simplified that so that there's one color. There's actually 560 of those in the genome. And we don't put in just one sensor, we put in 10 sensors. And so it's the combination of the readout that tells us what specific disease you have. It still won't actually, at the moment, tell us where it is, right? Because it could be still localized or it could have spread. So we have a second generation technology now where once the urine signal lights up, then you could go and do an imaging test, what's called a PET scan, and then you could find out where it is.
And then do you determine the type of cancer it actually is? And then they got particles
Yeah.
that's got an active site for that.
So we have a 10-plex, so these 10 different enzymes, these panels, and then we've developed them for different types of cancer. So we have a lung cancer one, we have a prostate cancer one, we have an ovarian cancer one, and a colon cancer one so far.
Thank you.
Sure.
How do you manufacture the nanoparticles? Like how accurate they are? Like what is it easy to manufacture?
Yeah, so the question was about nanoparticle manufacturing. And so I showed a bunch of different nanoparticles. The gold nanorods, for example, those are made in solution. And you use a surfactant on the surface so that they'll grow what we call anisotropically. So it's a solution process, and the polydispersity, which is how variable the size is very tight for that kind of particle. The polymer particles are much more polydispersed. So it really depends on which kind you're making. There have been recent studies where people use those microfluidic devices that I showed to actually try and make nanoparticles more uniform, because you could make them one by one.
Is the startup culture important for getting this new technology going?
Yeah, it really is. I think, so my first faculty position after leaving MIT was at UC San Diego. And I deliberately waited to come back to start my first company because I really wanted to be here in Cambridge in this ecosystem. I thought that would be the best thing for the company. And the reason to start a company is the best thing for the technology. Because when we create such early stage inventions that they really need to be grown up in a startup before they can be taken to the next level, whether they're acquired or to patients.
And has anything gone far enough that you can judge how physicians are picking up on these things? Or are they made some changes?
Yeah, so so Bob Langer, who's a great hero of mine, says it takes about 14 years to get a new invention into people. And I've been a professor 15 years now. I'm coming up on my time limit. The silver nanoparticles, the acne treatment, that's in patients already, but it's still in clinical trials. So we don't yet have anything where we can say it's fully adopted. The pharma tool is adopted, but it's a tool, not a product, not a clinical product.
So I have one question about your artificial liver. So I think the liver is made of many different types of cells.
Mm-hmm.
So how do you plan to assemble those different types of cells in a correct location?
Yeah, that's a great question. So there's this sort of philosophical debate in regenerative medicine and tissue engineering, which, you know, I welcome you to have a stance on. I'll tell you my opinion, which is how much biomimicry is required. Does it have to look like the organ with all of the component parts, or does it have to function like the organ? And so my favorite sort of analogy to this is, you know, if before flight was invented, if you looked at a bird and you saw this is how nature flies, maybe that's how you would have built an airplane. But in fact, that's not how we built the airplane. So we favor, for the liver, we favor the essential functions for life. And we've ignored a lot of things. So the macrophages of the liver, which are called Kupffer cells, are important for the immune function. And we don't have them in there. We have versions of it where we've added them, but usually we leave them out. The bile function that someone mentioned, we don't have a bile duct. So we've sort of chosen the subset of functions that we think are important for this class of diseases. But you can add them back and come up with clever patterning methods to try and study their role. About 70% of the cells in the liver are this particular cell type. So most of the other guys are smaller players. I don't know if I answered your question. Okay.
Any more questions? If not, I'd like to thank Professor Bhatia so much for joining us this evening.
Thank you.